Terrific. Well, good afternoon, everybody. I have the difficult position of standing in between your lunch. So I'm going to do my best to keep everybody excited about what we're talking about. And as Frank mentioned, this is actually the first presentation of our company. And so happy to share what we're working on. First off, we have a novel approach with the potential to more effectively treat multiple types of cancer without toxicity, inflammation, or off-target effects. We have a world-class science team that's led by Dr. Bert O'Malley. Dr. O'Malley is the Chancellor of Baylor College of Medicine. He's won over 65 awards, received the Presidential Medal of Science, and has been nominated for the Nobel on several occasions. They've published over 700 publications from the lab and from Bert O'Malley's work over the last 50 years, where he's affectionately known as the father of molecular endocrinology. And so Bert has spent his entire career focusing on genes and hormones. Baylor actually built him a lab over 50 years ago, and he's been focusing in this area with about 20 scientists over that period of time. We have a very strong partnership with Baylor. So the structure of CoRegen is that we've actually licensed the technology from Baylor, worldwide exclusive perpetual license, what you're going to hear about today. All of the science team that is within Baylor today is actually working for our company as well as Baylor. And so we have a very unique relationship with Baylor College of Medicine, which many of you may know is one of the top research institutions in the world. They own 25% of the company. They've given us access to not only Bert's lab, but also the extensive research and infrastructure at Baylor. So we have all of their infrastructure, saving us time, effort, and capital in pursuing what we're talking about today. Over $55 million has been invested over the course of the last 30 years on the science we're going to talk about today. We have very compelling preclinical results on multiple forms of cancer, and we've got a very, very strong patent portfolio and an extensive pipeline of applications. A couple of my colleagues have talked about this already. Cancer kills 10 million people a year, $1.2 trillion economic cost, and one of the scariest diseases that 40% of the world's population will face in their lifetime. Now, a little bit about cancer, some of you may know. The immune system cannot kill cancer if it can't detect it. And you'll see from this little chart here on the side, that T regulatory cell, that little red cell, is actually standing in the way of the immune effector cells. The immune effector cells, those cells that are designed to kill the foreign elements in our body, actually can't see the cancer and therefore can't actually penetrate and attack the cancer. So what we've discovered, our key discovery, is that within one cell, which is the T regulatory cell, we've identified one gene that when engineered, which is called SRC3, steroid receptor coactivator 3, when engineered, it allows the natural immune system to see and attack and kill the cancer. We've discovered that it's overexpressed in the majority of human cancers. We've identified that the Treg cancer protection activity is dependent on SRC3, and that by removing SRC3, the immune-driven response of the cancer is promoted. It fundamentally changes the immune system's ability to recognize and attack cancer. A little bit about SRC3, and I'll start by saying this is 50 years of research, 30 years on this family of genes. This lab, led by Bert O'Malley, actually discovered the family of genes 30 years ago. So there is no group in the world that is more of an expert within this area of science. It is very complicated, and they've spent decades doing the research, and you'll hear a little bit about the outcomes today. The first is SRC3 controls the expression of checkpoint proteins. We've talked a little bit about that over the last day and a half. It regulates growth in cancer cells and immunity in Treg cells. It's the second most overexpressed oncogene and it's highly expressed in Tregs. And it's the master regulator of the genome. This particular gene is seen as almost like a master switch. It affects other genes within the cells where it's altered or engineered. It controls hundreds of genes. And as I mentioned, it was actually identified in the O'Malley lab in 1995. Another important thing is that we actually work within the nucleus of the cell. Many therapies that are both in the market and in development are focusing on the cell surface. We actually focus in the nucleus, which allows us to specifically alter the cell biology without some of the off-target effects that you see from altering the exterior or the cell surface. So we've seen our ability to change the cell biology only within the tumor microenvironment without affecting what the Tregs are naturally supposed to do within the human body. Now, this is just a depiction here. Simply by knocking out the SRC3 gene, this is some of the impact that's seen within the cell biology. Most of these genes are specifically or individually targeted by companies because they are affecting cancer in some way. And by removing SRC3, these genes are all, for the most part, suppressed and in some cases enhanced. We've also seen that our altered T regulatory cell, the engineered, where we remove SRC3, proliferate faster than normal Tregs and are actually more viable than normal Tregs. You can see here and here, you can see where our re-engineered Tregs are actually, again, proliferate faster and are more viable. Now here's our therapy process. When we actually inject this re-engineered blood, removing the SRC3 from the Treg, the Tregs, engineered Tregs, actually infiltrate the tumor environment. And again, keep in mind, the normal Tregs can't get in the tumor environment. These actually are drawn into the tumor environment. Once they're drawn into the tumor environment, the T effector cells, all those natural killer cells our immune system has, are actually drawn in, and between the re-engineered Tregs and the T effector cell, it actually destroys the tumor. Our memory cells, the memory cells are actually increased, and a smaller number of SRC3 knockout Tregs actually dominate the normal Tregs within the body. There's been a very robust validation strategy. So far, we've focused, it's in the mouse model. We've done dozens and dozens of studies over the years. We've now tested it in triple-negative breast cancer, advanced prostate cancer, pancreatic cancer, and glioblastoma, all using the exact same therapy and all achieving the exact same results across all of the different tumors. And we're now looking at others as well. And we've stress tested it. We've injected the re-engineered Tregs, then injected the cancer. We've injected the cancer, then the re-engineered Tregs. We've injected the engineered Tregs, then cancer, wait until it's eradicated, then re-injected the cancer without additional therapy. And again, consistently, all the results have been the same. And actually, in one study, we injected the re-engineered Tregs and waited almost a year to inject the cancer, and the cancer never grew within the mouse. We've also seen dose-response studies, which further validates the therapy that we've designed. Now, here's just a couple of, as I mentioned, we've done dozens and dozens of studies. Here's just a few of them. You can look here. These here are the control mice, and these are the mice that have the Treg or SRC3 knockout Treg. And you can see all of these mice here have to be sacrificed after 30 days. And here you see a little bit of tumor growth and then flatlined. And now this doesn't go out to the length of the life of the mouse. All of the studies that you're going to see today, the mice live normal, productive lives. So they live till old age and then die. Here you'll see the actual tumors that were taken out when the mice were sacrificed. These here were the tumors within the control. And this here is actually just scar tissue. There was no evidence of any cancer left in the mice. We've done the same thing here. You can see this is advanced prostate cancer. The control mice all see accelerated growth of the prostate cancer. And then here you see it actually never grew with those mice treated with the engineered Tregs. Same thing with glioblastoma. All these charts are going to look relatively similar. You see the control mice here, sacrificed within 30 days. And here the tumors never grow. And again, these mice live their full lives. And we've also tried with pancreatic cancer with similar results. You see extremely aggressive cancer. The tumors grow rapidly. And here in the treated mice, the tumors don't grow at all. This is a little more aggressive study that I mentioned briefly. Here you'll see the breast cancer was injected in the mouse. You'll see the tumor never grew. We waited 7 weeks and then re-injected the cancer, but not the therapy, and the cancer never grew, which just speaks to the concept of increased memory cells that have been created and the longevity of the therapy that we're actually administering to the mice. And we've seen consistent results, and I have mentioned some of them already. Eradication of aggressive cancer cells, eradication, no remaining element of the cancer in any of the studies that we've actually performed. There was no recurrence in all of the studies that we've done, speaking again to the longevity of the therapy. It doesn't inhibit other critical functions, and I mentioned 2 areas that are relevant there. One is all of this activity, this change in cell biology, only seems to happen within the cancer microenvironment. And when it's outside of that, the normal Treg functionality is allowed to continue. So the cells look generally similar. We've seen 0 signs of toxicity and inflammation. So we've looked at every, the spleens of the animals, et cetera, and we've seen no inflammation whatsoever. And we've seen no off-target effects by any of the therapies that we've administered. Normal animal reproduction, they live their full lives, normal litters, and so we've seen no changes in the mice that have been treated by the therapy. Now, why our therapy works, I'm not going to step through all of this, but I just want to summarize all of it because this is very new, this is very complicated, and it's decades and decades of research that really hasn't hit the public, right? I mean, most of the publications, if not almost all of them, have come from the lab or our scientists, right? So that's really where the research has emanated from. Targeting SRC3 in the nucleus enables broad changes. So if you remember that little chart, suppression and enhancing of different genes, it really affects a broad list of the proteins that affect the body's immune system's ability to identify and kill cancer. It does block the Treg's normal blocking of the T effector cells. So it actually prevents them from blocking the treatment of the cancer. And then the engineered Treg, as I mentioned, they functionally dominate normal Tregs. They only act like this within the tumor microenvironment and not outside the microenvironment. And the next several things just talk a little bit about the fact that these natural killer cells are actually pulled into the cancer and the tumor. So not even if you were to take a look at how they would normally act, they're actually drawn even further through some of the increases in these different proteins, et cetera. And increased T memory cells, and again, no cytokine storm whatsoever and no apparent toxicity in the animals that have been tested. A little bit about our vision for how we would administer this. It's expected to be an outpatient treatment. Go in as you would a blood transfusion. Blood comes out. The Tregs are separated out. They're re-engineered. Your blood is put back in your body. And then the therapy is expected to be a fairly quick turnaround within 30, 60, 90 days. So we do expect a quick turnaround or a quick proof of it being effective. Now, the other thing I would note is this is the ex vivo approach. We have a number of, I would say, very, very creative and unique approaches in order to administer this in vivo. But this will certainly be our first approach to testing out the therapy in the commercial world. A little bit about our roadmap to commercialization. First thing is we are going to initially focus on triple-negative breast cancer. We expect to be in phase 1 next year. So we've done an enormous amount, as I mentioned, an enormous amount of studies. We have tons and tons of data, but we still have things that we need to do in order to have a fulsome submission to the FDA. And interestingly enough, even though the science is very new and unique, there are elements of it that have already been approved by the FDA. Most of you have heard of CAR-T, for example. There are elements of our therapies that are similar, which should make our pathway through the FDA even smoother. And having said that, we've surrounded ourselves with some of the top FDA folks, so we expect to be able to move through the process here. Just a little bit about our pipeline. So there's two elements here. One is we've talked about cancer. It's our main focus. It's our first focus. You'll see the list of cancers we plan on focusing on. And then the other thing that I'll mention is that we do have several patents. Our main patent was just issued 8 weeks ago, actually. That patent covers us for all cancers, all forms of editing the Tregs. And actually, during the process, there was no prior art objections, which means that they couldn't find any other therapies or submissions that were remotely like ours. There are two other patents here that I haven't really talked about, and that's around the small molecules. And interestingly enough, prior to the cancer discovery, the lab had seen tremendous results of modifying, either enhancing or inhibiting SRC family. There's 1, 2, and 3 parts of that family. We saw incredible results in post-MI patients, 55% reduction in cardiac scarring, and in post-stroke, we've seen 65% reduction in recovery, or in some of the complications. So there are a whole list of expanded applications. There's only so much that we can do. We've had the license with Baylor for 6 months, so we've been very, very busy, as you can imagine. But we think that we've got an incredible pipeline of opportunities. This is truly a platform leveraging decades and decades of a brilliant scientist that's been within a lab, and our job is to bring him out of the lab and commercialize it for humanity. So a little bit about the team. I've spoken about Bert O'Malley. Again, please Google him. He is just an astonishing individual. The team you see below, they've all known Bert ranging anywhere from 25 to 45 years. So it's a team of people that have worked together for quite a long time. In addition to that, we actually have 18 other both scientists and technologists that are working with us full-time, again, split between us and Baylor, but all on the same subject matter and working on our therapy. Now, there was an interesting comment someone made yesterday when there was the presentation about the public relations efforts and how to really promote what you're doing as a business. And I think that the comment was, scientists are typically introverts. And what you'll notice over time is that universities, which have tremendous intellectual property, in a lot of cases aren't able to commercialize that. And there are all sorts of efforts in order to do that. So we've put together a team that not only knows the science, which I'm not a scientist, I'm a business guy and mostly a tech guy, but I've spent a whole bunch of years in healthcare as well. But we want to make sure that we've got access to capital. We want to make sure that we can tell the story the right way, know who to tell the story to. We can come up and overcome any objections, because I wish that companies became successful because of their science. It's not just the science. It's the ability to take that science and commercialize it. So Steve Gorlin, our executive chairman and CEO, I've known Steve for 20 years. His whole career has been identifying science from universities and commercializing it. He's had a number of successes, 6 major ones, 4 that have reached over $1 billion, 1 over $2.5 billion, and the last one he founded was a company called Rejuvenation, which was sold to Pfizer for about $14.2 billion. Myself, 30 years, technology, healthcare, finance. I've built companies. I've been director, advisor. I've raised billions of dollars of capital in my history. John Thomas and Steve have worked together for 40 years. John's been the CFO for public-private companies of all sizes, and Keon is a marketing and branding wizard. And so we want to make sure that we've got the business side, not just the science side, supporting the company. Now, from a board perspective, as we have started to release this to the market, almost every significant scientific brain that we've interacted with has been blown away by the science, and several of them have asked to get involved. Frank Prendergast. Frank was the head of the Mayo Clinic Cancer Research Center. He was on the board of Eli Lilly for almost a decade. You have Minnie Baylor Henry, who was at the FDA for a decade and then went on to run Johnson & Johnson's regulatory practice, bringing products to and through the FDA. We have an advisory board. And by the way, again, we're only 6 months having gotten the license, so we are going to be expanding both the board and advisory board. Roy Herbst just joined us recently as the chief of medical oncology at the Yale Cancer Center. Mark Siegel, very, very well-known vocalist, affiliated with NYU Langone, and Scott's a financial advisor for us as well. And just to summarize here, again, hopefully through the presentation, you've seen it's a very, very novel approach. It's incredibly exciting. There is nothing like this in the marketplace today. To be able to go after multiple forms of cancer with the same therapy, no toxicity, no inflammation, that's our hope. That's the promise for the future. Very well-protected patent portfolio, no prior art objections, and quite a robust pipeline of opportunities and other therapies. Strong partnership with Baylor, which again just gives us access literally to all of the infrastructure of Baylor. So again, saves us time, effort, and capital. $55 million has already gone into the research over the last several decades. Preclinical results, I think, speak for themselves. We've got rosters of those sorts of studies if folks are interested. And so with that, I would certainly like to open it up to questions, since there's a big red zeros there. In fact, I've run out of time and hopefully kept your interest.